Welcome, dear friends. We are going to take a trip together through a very special experience. Today, my generative model and yours will connect. I'm going to activate my GPT-like generative model in my head to describe a deep journey from the outside of the brain to the microtubules inside the axons and dendrites of neurons, drawing and explaining parallels between all of that and artificial intelligence. And you and us all together are going to visualize that journey with our generative models. This will be a journey with analogies, with metaphors, and with concrete data too. A journey exercising and flexing the generative model of your brain to visualize this adventure, actively exploring our biology with all its electrochemical processes and those comparisons and parallelisms between that biology and artificial intelligence in order to engrave deeply in our minds where both systems match and where they differ. So I invite you now to silence your mobile phone so that you can immerse yourself deeply in this experience where you are going to use your own cortical columns to visually generate those very same cortical columns in a wonderful, recursive visualization and loop where we all meet. And for this, you have to make a proactive effort in this experience to really try to visualize what I am describing to you, to exercise and activate your generative model, because your generative model is the one that is going to produce a visual interpretation in your mind of this journey. A visual interpretation that will help you to record and preserve this journey in deep and lasting ways. And we don't look for absolute precision, whether we have 100 billion neurons in the brain, or if they are 86 or 95 billion. We will use approximate numbers, as precise as possible. But the important thing are the general concepts and the multidimensional journey to the center of the neuron as we connect it all with artificial intelligence. And we are going to go all the way from the outside of the brain to the microtubules inside the axons and the dendrites of neurons. And now, come with me, my friends, come with me, and let's begin the journey. Imagine a solid container made of bone. It is your brain, and you Imagine it, you visualize it now, floating in the middle of space. A solid container made of bone, the size of your head, which contains inside a salty ocean. It is your brain. That container made of bone, the brain, is made up of several parts or lobes. The frontal lobe in the frontal part, the occipital lobe at the back, the parietal lobe above, and the temporal lobe below. And within this container that you are visualizing now, the part that occupies 70% of its volume, the part that is the most evolved one, the one that has evolved most recently, is our neocortex. That neocortex, surrounded by that salty ocean, is full of tissue, cells, and minerals. And within that salty ocean, among all that tissue, we have what we call ions. What are ions? Ions are atoms whose positive and negative charges differ. Atoms that have an electric charge, positive or negative. These ions are, for example, potassium, sodium, chlorine, and calcium. That's why these minerals are so important to us and our brain. Sometimes you hear the word electrolytes. Electrolytes are minerals in the body that have an electric charge. And later we will see why these ions are so important. But in this salty ocean, we also have all of that tissue, many cells. And how are those cells distributed? And what do they look like? And how do they work? So we are all of us right now visualizing that container made of bone, filled with a salty ocean, and a lot of tissue and minerals inside. At the moment, you visualize it in a diffuse way that will become more and more clear as we zoom inside. So now, let's zoom inside into our neocortex, all right? So if we zoom into the neocortex through that container made of bone, we find that area, the neocortex, which occupies 70% of the total volume of the container. And there we find 150,000 columns, vertical columns, of tissue and cells, which are in a way like 150,000 spaghetti. What? Let me explain. The famous American neuroscientist Vernon Mountcastle discovered the vertical columnar structure of the human neocortex. The neocortex is divided into a number of vertical columns, about 150,000 in total, which you can see as if they were 150,000 spaghetti, because if you consider the size of your head and you try to put 150,000 vertical columns inside, you will realize that each of the columns will be very thin, like spaghetti. So you are now visualizing these 150,000 vertical columns, thin columns, like spaghettis. Each of them has a lot of tissue and cells. And they are inside of this container made of bone. Each of these columns, the cortical columns, has about 100,000 cells, which we call neurons. And each column also has micro columns, and each micro column has several hundred neurons. So think of each column as a circuit, as an electrical circuit, a kind of a chip or a processing unit, a circuit that is repeated many times throughout the neocortex. And the microcolumn would be like special purpose processing unit. So now you are visualizing those 150,000 cortical columns, those 150,000 spaghetti, in a diffuse way, in your own way. It doesn't matter in which way. The important thing is that you are right now exercising actively and proactively, flexing your own generative models. And soon we will begin comparing parts of this biology with artificial intelligence. But now, let's zoom in again inside one of these cortical columns, inside one of these 150,000 spaghetti. Because you see, each of these columns, each of these spaghetti is made up of 6 layers vertically, which we name from top to bottom 1, 2, 3, 4, 5, 6. From top to bottom, the 6 layers of each vertical cortical column. And within each of these 6 layers, we find cells, neurons, and one of the most common types of neurons in the neocortex is the pyramidal neuron, which the Spanish Nobel Prize Santiago Ramón y Cajal drew so wonderfully. Santiago Ramón y Cajal created these beautiful drawings of pyramidal neurons that are so beautiful that they are still being used today to illustrate articles on neuroscience and the like. So well, my friends, let's visualize together a pyramidal neuron. Each pyramidal neuron is made up of a body that we call soma. Also some inputs that we call dendrites, from the Greek dendro, which means tree, and that are like branches of a tree, like twigs of a tree, and an output which we call axon. The body is soma, the inputs dendrites, the output axon. The body, the soma, has a pyramidal triangular shape, and that is why we call this neuron a pyramidal neuron, because of the shape of its body. So now you are imagining that little body that is like an elongated pyramidal triangle. Imagine the vertex, the single vertex of the triangle pointing upwards and the base of the triangle below. Now, the inputs that carry signals to the neuron are called dendrites, from the Greek dendro, which means tree, because they are like branches on a tree. And we have two types of dendrites in the pyramidal neuron. We have the basal dendrites, basal from base, which arise from the body of the neuron, from the bottom and the lateral sides of the body of the neuron. We visualize them now. You visualize them as little branches of a tree that arise down and to the sides of the body of the neuron. And then we have the apical dendrite, a vertical branch that rises from the body of the neuron upwards, proud and vertical. And from that apical dendrite, other branches, other twigs emerge at its sides, small twigs, more numerous at the very top, at the top of that apical dendrite. Therefore, multiple basal dendrites below and one proud vertical apical dendrite that rises upwards from the body. So you are now visualizing that pyramidal neuron, the little elongated triangular body, the basal dendrites sticking out from the body down and to the sides, like twigs on a tree, and the apical dendrite sticking out proudly from the body upwards, having twigs itself that arise from it, especially in its upper part. And then the neuron has an output, the axon, which is like a strong and proud branch that comes out of the body and connects with other neurons. The axon, the exit, the output. And soon we will see how it works and where it goes. How beautiful is the pyramidal neuron, right? You are visualizing it now. And after this experience, I will invite you to explore the wonderful drawings of these neurons by Nobel Prize Santiago Ramón y Cajal. So well, we have about 100 billion neurons in the entire brain, and about 16 to 19 billion neurons in the cortex. And each of these neurons connects to several thousand other neurons around it. But what are the dynamics of the biological neural system of ours? How does it behave? And how does it learn? And how do some of its characteristics compare with artificial networks? Visualize with me the pyramidal neuron. The neuron has a membrane, which is like a thin layer of fat, lipids. A thin layer of fat, the membrane that separates the exterior from the interior of the neuron. In that membrane, there is a voltage, which is the difference between the electrical charge on the outside and the inside of the neuron. We measure this voltage in millivolts, thousandths of a volt, because the brain uses very little electricity. It is very efficient. It uses about 20 watts of power. It's equivalent to powering very weakly a light bulb that shines very dimly. This is already a difference between our biology and artificial intelligence, because a modern NVIDIA GPU graphics card consumes hundreds of watts of power, between 350 and 550 or more watts of power, one single GPU, while the entire brain is consuming only about 20 watts of power. The brain is much more efficient, and soon we will see some of the reasons why this is the case. And there is a lot of active work nowadays in artificial intelligence to try to make artificial networks more efficient, because right now, in a way, artificial networks are brute forcing their way to their incredible results. But let's go back to the membrane of the pyramidal neuron. You are visualizing the pyramidal neuron, and remember that around the neuron, in that salty ocean, there are those ions, those electrically charged atoms. The membrane of the neuron does not allow those ions to pass through it. However, in the membrane there are some proteins that build little channels that are what we call ion channels. Ion channels through which the electrical current can flow, and that selectively allow certain types of ions to pass. So now you are visualizing the body of the neuron with its fatty membrane that has little channels in that membrane that can let the electrical current and those electrically charged atoms, those ions, to flow through. And they can let those ions pass to, for example, balance the voltage in the membrane when necessary. When a lot of voltage is created and we need to, for example, rebalance it again, this passage of the electrical current can be allowed. And we will see why and how it works in a moment. But what a beautiful and efficient system we have. Some minerals that have electrical charges that help us regulate the voltage we have in the membrane of the neuron. So now, again, visualize that membrane of the neuron around the soma, around the body of the neuron. And listen, the resting voltage of a typical neuron is around minus 70 millivolts. That is, when the neuron is not active, when it is not sending signals through its axon, then it has that voltage, minus 70 millivolts. But if that voltage goes up and it reaches a certain threshold around minus 50 or minus 55 millivolts, typically, then the neuron fires what we call a pulse, a spike, an action potential that we call it. A pulse that travels down the axon, and we will see where it goes. So you are visualizing the membrane of the neuron, and the voltage that rises beyond a threshold, and then a pulse is fired, spike, an action potential, one bit of information that travels through the axon on its way towards other neurons. You visualize it. And the place most commonly associated with the initiation of one of these spikes, that again we call an action potential, is in the axon, specifically in the initial segment of the axon that we call the axon hillock, near where the axon joins the soma, the body of the cell, the body of the neuron. It is at this point that the neuron is most sensitive to changes in that voltage, and where an action potential is most likely to fire in response to incoming stimuli. Although other parts of the neuron, the soma and the dendrites, contribute as well. Now, keep visualizing the pyramidal neuron, because once a pulse, once spike, an action potential is fired, the voltage on the membrane is going to be rebalanced back to the resting voltage. So those little ion channels in the membrane will let current pass through in order to rebalance that voltage back to the resting voltage. And if we want to fire another pulse, another action potential, another bit, we will have to again push that voltage again to rise above the activation threshold. Aye, my friends, thinking requires effort and energy. It requires glucose. It is not so simple, because to trigger those spikes, to trigger those pulses, those action potentials at the neurons through the axons, you have to rise above, you have to exceed that voltage threshold. And that is why we have to eat well, to have that power, that glucose, to put in motion those spikes along the neurons and axons. And so, my friends, using the temporal dimension, the neuron puts in motion what we call spike trains, action potentials that follow one another, trains of pulses, a mixture of pulses and pauses. Pulse, silence, pulse, silence, pulse, pause, pulse, pause, spike, pause, spike, pause. A train of spikes that encode information through the temporal dimension. If we could listen within the axon, we would hear something like so now you visualize those trains of bits, trains of pulses, trains of action potentials that travel through the axon of the neuron. And this is another important difference between biology and artificial intelligence. Our biological neurons take advantage of the temporal dimension, encoding information within trains of pulses that use that temporal dimension. Typical artificial intelligence does not use such temporal dimension. When we make inference and feed information into an artificial neural network, the computations occur progressively through the layers of the artificial network, but all at once, progressively from input to output. So the typical artificial network, we are talking about the typical one, still does not take advantage of the temporal dimension. But there is a lot of work and research on things like spiking artificial neural networks that are using that temporal dimension, but are still far from working as well as typical artificial networks. So let's continue. How does the interface work, the connection between the inputs and outputs of different neurons? Visualize now several of these pyramidal neurons, many of them with their bodies, their dendrites, their axons, and their axons connecting to the dendrites of other neurons, to the inputs of other neurons. But how do they really connect to each other? Well, you see, the pulses, the spikes that travel through the axons arrive in the final part of the axon at some terminals where there are some vesicles, some sacs that contain neurotransmitters, which are chemical messengers. So the end of the axon is not directly connected to the dendrites of other neurons, to the inputs of other neurons. There is a gap. There is a gap between the axon and the dendrites of other neurons. That gap is called a synapse, which comes from the Greek point of contact. The synapse is the gap through which the neurotransmitters that go from the axon to the dendrite travel. So the neurotransmitters are released from the terminals at the end of an axon, and they travel through the gap, through the synapse between an axon and a dendrite, and arrive to the dendrite. And these are chemical messengers, the neurotransmitters, that are going to take care of exciting, inhibiting, or modulating the neuron to which they arrive. And on the other side of the gap, the dendrites in each of the branches of the postsynaptic neuron, they have little spines that contain receptors adapted to receive certain types of neurotransmitters. And again, these neurotransmitters have the function of either exciting, inhibiting, or modulating the neuron to which they arrive. The best known excitatory neurotransmitter is glutamate. The best known inhibitory neurotransmitter is GABA. And other well-known neurotransmitters that modulate neurons are dopamine, serotonin, acetylcholine, and norepinephrine. So these are all neurotransmitters: glutamate, GABA, dopamine, serotonin, acetylcholine, and norepinephrine. So visualize now, visualize the end of an axon. The signal, the spike, travels and reaches the end of the axon, and it reaches some terminals. And at those terminals there are these vesicles, these sacs. Inside they have neurotransmitters. When the signal arrives, those vesicles, those sacs, release the neurotransmitters into the gap, and they travel through the gap and they reach the receptors at the spines of the dendrites of other neurons. They reach the receptors that are adapted to receive certain types of those neurotransmitters. And I would like to remind you again about the difference between the ions that we talked about before and the neurotransmitters that we are talking about now. Ions are individual atoms or molecules with an electrical charge. Neurotransmitters are more complex molecules that facilitate the communication between neurons by being transported across these gaps. So well, visualize with me when a strong pulse, when an action potential spike reaches the end of the axon, the contents of these vesicles, these sacs, are released. The neurotransmitters are released and they cross the gap and reach the receptors of the dendrites of other neurons. So now, neurons therefore receive temporally encoded information through their dendrites, through their inputs, and integrate that information to then proceed themselves to excite, inhibit, or modulate other neurons when the voltage across their own membranes exceeds the necessary threshold, and so on and so forth between multiple neurons. And here we are going to establish another very important parallelism between biology and artificial intelligence. Let's see. The artificial neuron receives inputs from other neurons, multiplies each of its inputs by a weight, a small number that denotes the strength of that connection, of each of those connections, and sums all of those computations. And after that, it passes that result through an activation function, which is like a threshold that, if exceeded, allows the signal to pass and otherwise sets it to zero. So well, the process that we visualized before with the biological neuron of the voltage threshold that has to be exceeded in the membrane of the pyramidal neuron, we can consider that is somewhat analogous, similar to the threshold that the signal of the artificial neuron must also overcome in its activation function. For example, in its ReLU activation function located at its output. So, therefore, we have a somewhat similar analogy between the threshold at the biological and at the artificial neuron. The threshold that both of them have to overcome at or in order to generate the output. But now let's look at the inputs, because that's where things change. Because we used to think that the biological neuron in its dendrites did something similar to the artificial one, that it simply added, summed, integrated the signals that it received, taking into account also a weight, in this case, in the biology case, the so-called synaptic weights, which denote the strength and the efficiency of the connections within the synapse. So, somewhat analogous, but in a more complex way, because those biological synaptic weights depend on many parameters of the biological neuron, but in any case, somewhat analogous. However, my friends, in recent years, researchers have been discovering that the dendrites of human neurons are much more powerful than we thought. That they are by themselves, the dendrites of the neurons, they are equivalent to a powerful non-linear computing system. Therefore, equivalent not to one artificial neuron, no, but to a whole artificial neural network with a few layers. To be more precise, recent academic research showed that one biological pyramidal neuron was roughly equivalent to an artificial convolutional network of between 5 and 8 layers. Incredible, right? Incredible. Biological neurons are much more powerful than artificial ones when we compare them one to one. And how is it possible that our dendrites are so powerful? Well, in a very simplified way, researchers have discovered that dendrites, the inputs of neurons, are capable of, among other things, emitting their own pulses, their own action potentials that we call dendritic spikes or dendritic action potentials. Their own action potentials, their own pulses of different types to stimulate the membrane of their own neuron in many different ways. So they can emit pulses of many kinds. One of them, for example, NMDA spikes. Another one, calcium spikes, many types. Different localized events in response to different input patterns. So, that is, when before we talked about the action potential, the spike, the pulse that travels through the axon to stimulate other neurons, that output that is activated when the threshold is exceeded at the membrane, at the axon hillock, that action potential is the main one at the neuron, but not the only one, not the only kind of pulse of action potential that happens in a neuron. It is not the only type of computation and pulse in the neuron. Instead, at the inputs, at the dendrites, there are other types of pulses that are produced and which in combination with each other allow those dendrites to behave like powerful nonlinear computing mechanisms. And that is what makes the biological neuron as a whole equivalent to, for example, in that recent study, an artificial convolutional network of between 5 and 8 layers. Incredible, right? Therefore, at the output, the threshold side of things, at the output of the neurons, is quite analogous between biological and artificial neurons. Obviously, saving the distance in terms of complexity, because the biological neuron is always way more complex than the artificial one, but somewhat analogous. But however, at the inputs, this is different. The inputs are not so equivalent. The inputs are much more powerful in the biological neuron. Now, however, in terms of processing speed, the biological neuron is slower and the digital artificial neuron is much faster. A biological neuron typically fires a pulse, an action potential, a few times per second, like between 10 and 200 times per second. For example, they can fire every 5 milliseconds or 200 times per second. But artificial neurons can be much, much faster. But in contrast, biological neurons can operate in massively parallel, simultaneous fashion, which artificial GPUs can also do. Artificial graphic processing units, GPUs, can work in parallel and can compute with those neurons in parallel, but doing such parallel processing at the scale of the human brain would require an energy efficiency and computing power that we have not quite reached yet. So the parallelism of the human brain, with billions of neurons working simultaneously with tremendous energy efficiency, is still out of the reach for artificial networks. And this is where we can see that if we continue to improve and evolve our AI architectures, together with the power of the hardware that we have, AI has a lot of room for further improvement ahead. Okay, now you have visualized a lot of the process of how the neuron works. But now we're going to enter the last third of this journey. In the last third of this journey, let's explore how does learning occur. Learning. Both biological and artificial learning is based on tweaking, modifying the weights of neural networks. In biological networks, synaptic weights express the strength and the efficiency of the synapse, those gaps between axons and dendrites. How effective they are in passing, in transporting signals from one neuron to another. That's what synaptic weights express. In artificial neurons, weights are numbers that express the strength of the connections between the digital neurons. Now, in biological neurons, learning is very closely related to what we call synaptic plasticity. When two neurons or a pattern of neurons are activated at the same time, their synaptic connections, their synaptic weights are strengthened. They become stronger. It is Hebb's principle. Hebb's principle says, neurons that fire together wire together. Neurons that fire together wire together. As neurons become active at the same time, the strength of their connections with each other increases. And this connects with two key concepts in relation to synaptic plasticity: LTP and LTD, long-term potentiation and long-term depression. If we repeatedly and rapidly activate certain neural patterns, we strengthen those synaptic weights and make it increasingly easier to raise the membrane voltage above the critical threshold for the neuron to fire a signal through the axon, and therefore changing the strength of the different synaptic connections. We are learning and creating habits. If we keep repeatedly and quickly activating that certain neural pattern, we strengthen the synaptic weights, therefore we improve the efficiency of that transmission of the signal, and then we make it easier to raise the membrane voltage above the critical threshold for the signals to be fired, changing the strength of the different synaptic connections, and so on and so forth. And that way we are learning and creating habits. There are still many unknowns about how this learning develops. LTP and LTD, long-term potentiation, and the opposite, long-term depression, are some of the mechanisms involved in learning processes and habit creation. But there is much more that would take us many days to comment. Now, in the artificial side of things, we mainly use mathematical computations that make use of the gradient descent and backpropagation algorithms, which have proven to be very efficient so far. When we train an artificial network, neural network, we compute an error signal at its output, an error signal that expresses the difference between what we are getting at the end of the network and what we want to get, what we produce at the end of the network and what we want, what we would like to produce, that difference, that error. and then we reverse propagate that error signal backwards through the network. That's why we call it the backpropagation algorithm, and we use that error signal to calculate the partial derivatives using mathematical calculus, the partial derivatives of each of the weights of the neurons with respect to the output of the network. And this mathematically allows us to know in which direction we should modify each of the weights to help reduce that error signal, that error at the end of the network. If we now combine that with the gradient descent algorithm, what we are going to do is to gradually, iteratively, modify each of these little numbers, each of these weights, so that gradually the output of the network produces a smaller and smaller error signal. Gradually the error gets smaller and smaller, so the performance of the network gets better and better, and the network is learning. So what we are doing is, in a loop, gradually, iteratively, we are using the error signal at the end of the network and using the chain rule of calculus, of mathematics, to calculate the partial derivatives of each of the millions of weights of the neurons of the network in relation to the output of the network and to that error, to understand in what direction do we need to push each of the weights, each of the millions or billions of weights of that artificial neural network, in which direction do we have to push it a little bit to gradually decrease that error at the end of the network. And we do this over and over and over and over, and that error goes down and down and down until the performance becomes very good, the performance and the error becomes very small, and then the network has learned. But learning in biological networks is even more complex than in artificial ones, since the learning involves not only the changes in the synaptic weights, but many other processes, such as the possibility of creating new neurons, brand new neurons, or pruning connections, eliminating or pruning connections, etc. So we can say that biological networks have synaptic and structural plasticity of many types, while the typical artificial network presents plasticity mainly in its weights, in the strength of its connections. And now, my friends, how does the brain do planning? How does it decide what action to take? To plan, my friends, the brain needs to anticipate the future. It needs to predict what we call counterfactuals, things that have not happened yet. And for that, you need what we call a world model, a model of the world. What is a world model? Let's say the information that surrounds you and all of us, all of that complexity does not fit into our brain. So the first thing that a creature like us needs to do is to compress all of that information. But even more important is, how do you compress it? In what way you compress it? Because that complexity, all of that detail around us, contains a lot of fluctuations, a lot of noise, a lot of details that don't tell us anything useful. Such as, for example, that a certain point of light has 157 of red intensity. That is not useful. What we really want is to abstract reality, compress it, convert all of that detail, all of that high dimensionality into a much smaller number of factors or dimensions that describe that reality at a much more abstract level, but that has useful meaning and that allows us to generalize and transfer that knowledge. couple of examples of what I mean by this abstraction. A map of Europe is an abstraction of the surface of the real Europe, and it is more or less useful as a map depending on how well you abstract that complexity of the actual surface of Europe. Typically, you want to create a map that is as simple as possible but as accurate as possible. Another example, if you consider millions of human faces expressed as images of 1000 times 1000 pixels, that is 1 million dimensions, 1000 pixels horizontal, 1000 pixels vertical, 1 million pixels, 1 million dimensions. Imagine that we compress that space of 1 million dimensions that expresses all the possible images of 1000 times 1000 pixels. We compress that space into just 200 dimensions, just 200 numbers, 200 factors. And we want each of those 200 numbers to encode at an abstract level something that gives us very relevant and useful information. For example, whether a face has glasses or it doesn't have glasses. What is the style of the hair? If the face is smiling or is not smiling. So that's what we do, you see. Those million dimensions had a lot of information that was not very useful for us, but now we compress it all into 200 dimensions, 200 numbers, but one number is going to tell us if the face has glasses or it doesn't have glasses. Another number is going to tell us if the hair is blonde or is not blonde. Another number is going to tell us if the mouth is smiling or is not smiling, etc., etc. So it is about compressing efficiently and in a relevant and useful way. In the same way, our brain, since we are babies all the way to adulthood, our brain is learning and generating models of the world, of objects, but also of concepts, of places, of emotions. They are all simplified representations of how the world works. That is a world model, a simplified representation of how the world works. And we could say that what separates an Albert Einstein or a Marie Curie from other people is how good are those mental maps, those models of the world, those simplified abstractions of how the world works, of concepts, of things, of emotions, of everything. And there is a famous theory of scientist Jeff Hawkins called the 1,000 brains theory that tells us that each one of those 150,000 cortical columns, and you are now visualizing again those 150,000 cortical columns, those 150,000 spaghetti, and you visualize one of them, and the 1,000 brains theory tells us that each one of them is creating different models of the world. That is, we don't have one single model of the world. We have many that are being created dynamically, that express different angles and perspectives and abstractions of whatever you are touching or looking at, that coffee cup that you are picking, that person you are looking at. And why is this useful? And how is it possible that we have a stable perception of reality and of things if we have so many internal models of the world? And how does this compare to what's happening with artificial intelligence? Does ChatGPT have a model of the world? Let's see. This is all fascinating, right? We are entering the last part of this journey. You are right now using your own cortical columns to reflect on themselves. Incredible, isn't it? So listen. Some neurons in the neocortex connect not only up and down, but also laterally with other columns. So I want you to visualize now different cortical columns. Each cortical column has a lot of pyramidal neurons, and the pyramidal neurons are connecting with other neurons up and down, but also some of them are connecting laterally, horizontally, with other cortical columns. And these special neurons are the ones that are thought to be involved in a process of consensus, of voting or integration, similar to the ensembling technique in artificial intelligence. And so in biological neurons, the hypothesis states that some neurons that are responsible for high-level concepts, they vote to create a stable consensus, a stable perception, sensation, and consensus. And in this way, even though individual neurons may differ or change, the stable perception that results from this consensus mechanism and voting is maintained. Amazing, right? In artificial intelligence, ensembling is a technique that we use in complicated projects. What we do is we use different artificial architectures and networks that reach different conclusions, and then we combine those conclusions to create a consensus. Well, something like that may be happening in the biological neural networks according to the 1,000 brains theory of Jeff Hawkins. And why is all of this so very useful? Because being multimodal and multimodal creates a very resilient system. We have a multimodal system which processes multiple sensory modalities using similar circuitry, similar processing units, and which is also multimodal because it creates multiple models of the world, multiple abstract perspectives of what we observe. And this produces a very resilient system in which even if some cortical column is injured or one of them changes locally, the high-level consensus is maintained. And does ChatGPT or GPT-5 or GPT-6 or GPT-7, does it have a model or multiple models of the world like our cortical columns do? Well, nobody knows yet. And there is a lot of controversy around it right now. There are some people that say that no, ChatGPT or GPT-4 or 5, they cannot have a model of the world similar to ours in their mathematically compressed, latent, abstract spaces. There are other people that say that based on the answers of the GPT systems, based on their capabilities, they probably have to have some kind of model of the world internally. A recent academic study based on the board game Othello seems to suggest that yes, that large language models, LLMs, probably build some sort of simplified internal representation of how things work. So the consensus is that probably large language models, LLMs, they probably have some kind of model of the world, some kind of rudimentary, maybe weak internal world model, still not very robust. That's why it's quite easy to make it fail. That's why when you use GPT, ChatGPT or GPT-4 or GPT-5, sometimes it hallucinates, it lies, it contradicts itself, because it probably has some internal model of the world, but is not very robust. For systems like GPT, ChatGPT, GPT-4, GPT-5 to have true, robust world models, as powerful as ours, we need to improve still some key parts of these systems. A robust world model is a model that is capable of predicting the next state of the world around it. It's also capable of predicting the consequences of performing a series of actions. It's also capable of filling and completing incomplete perception, incomplete information in our perception. It is capable of this and much more in a consistent and robust way. And for this to happen, we will have to continue evolving the architectural paradigms of artificial intelligence. And we could talk about that for many days, but we don't have time in this experience. Now, we are in the final part of the journey. The other point to highlight again is the great energy efficiency of our brain. Remember, we said that all the wonders of your mind are produced by the brain with just 20 watts of power. And why? How is it possible? Among other reasons, this is because of what we call sparsity. Sparsity in the amount of neurons that are active at any one time in the brain, but also sparsity in the network itself, in the way the network and the neurons connect to each other. You see? In terms of activation, only 0.5 to 2% of your biological neurons are active at any given time. Most neurons are not active at a certain moment. Only 0.5 to 2% of your neurons are active at any one time. This is a lot of sparseness, and this saves a lot of energy. On the other hand, in artificial intelligence networks, a large part of the neurons are active at one time. Typically at least 50%, at least. Some of them are deactivated by the thresholds of their activation functions, but typically at least 50% are active. So compare that. Only 0.5 to 2% are active in your biological brain, and at least 50% in the artificial networks. That's one of the reasons why artificial networks spend more energy. But again, not only we have biological sparseness in terms of neuronal activation, but also in terms of interconnectivity. The connections, because each biological neuron connects with only 1 to 10% of the surrounding neurons. On the other hand, artificial neurons typically connect with each other in very dense networks, very dense interconnections between the neurons. Not always, but many times. So biological neurons connect to only between 1 to 10% of the surrounding neurons. Artificial ones have typically more dense interconnection. And the point is that this biological sparseness makes the system very resilient and efficient, since each neuronal pattern involves a limited number of neurons, which makes them very resistant to noise, to injuries, and other problems. It makes the identification and conservation of those patterns way more resilient and more efficient, as sparseness means less energy consumption as well. We are reaching the end, my friends. I want almost at the end to do the last zoom for us to enter those dendrites and those axons of neurons. Within those dendrites and those axons, we find some structures that we call microtubules. These are fine, very, very fine tubes, cylinders that are responsible both for maintaining the shape of these structures of the dendrites and axons, and for helping to transport molecules and materials through them. And one of the most recent theories about human consciousness, created by Nobel Prize Roger Penrose and anesthesiologist Stuart Hameroff, tells us that according to this theory, human consciousness may be generated within and in relation to quantum processes that develop within these tiny structures, the microtubules. These microtubules within the dendrites and axons of neurons. This is the ORCH theory. Orchestrated Objective Reduction Theory, a theory that postulates that consciousness originates at the quantum level within neurons, rather than the alternative typical view that it may be a product of simply the connections between the neurons. And this theory considers that the mechanism by which consciousness arises is a quantum process called objective reduction that is orchestrated by those tiny structures called the microtubules. It is just one example, one of the many theories about consciousness, about how consciousness is produced. Just one of them, but it serves us nicely. It's nice to use it as the final stop in this journey, in this process of zooming, making zoom from the outside of the brain all the way to these tiny microtubules inside the neurons. And is artificial intelligence, or will it ever be conscious? And does an AI need to be conscious in order to reach AGI, artificial general intelligence, or artificial superintelligence? It is a long topic that would take us a long time to discuss. We leave it for another time. But to finish, I want to talk to you for a moment about the future. Today's artificial intelligence produces impressive and very useful results. However, we are still at the beginning of the road. Because, my friends, to create intelligent artificial entities that have robust world models like our brains have, that are highly efficient in terms of energy consumption, as are our neurons, thanks to, for example, that sparseness at multiple levels that they have, that make use of multi-modular architectures like the one recently proposed by Yann LeCun, that are capable as well of processing information at different speeds, fast and slow, in connection with Daniel Kahneman's System 1 and System 2. Inference and learning, fast perception-action loops versus the slow search for new algorithms, which also constitute entities capable of perceiving and acting in an environment, entities capable of, with their powerful generative models, modeling themselves as mirrors of their environment, capable of, as Carl Friston says, combining perceptual inference and active inference in order to understand their world, to understand themselves within that world, and to be able to survive in it in an intelligent and active way. For all of that and more, my friends, artificial intelligence will continue its path, improving more and more its paradigms, its architectures, transcending and complementing the advantages that scale provides. Scaling that has given us so much success so far, but that has to be combined with other improvements and with evolutions of our architectural paradigms in order to eventually reach AGI, Artificial General Intelligence, or ASI, Artificial Superintelligence. Thank you very much for taking this journey with me, and congratulations for exercising your own generative models and doing this wonderful exploration of your biological neural networks while you were comparing them with artificial neural networks. Congratulations and thank you.